In this video, I'll be talking about bacterial growth, what are the things that are required for a bacteria to grow within the laboratory system, that means the culture. And then I'll be talking about the different kind of gene transfer, and I'll touch a little bit about conjugation as well. Okay, so bacterial growth. Okay, just let's draw a bacteria. Now suppose this is a bacteria, okay, and I want this bacteria to grow in a culture media within the lab. So what I'll do, I'll put them into a culture, so that like within this particular culture this bacteria will grow and it will thrive. Okay, so either I can put it in a flask, so in that case this media will be a liquid one in which the bacteria will be grown, or else I can also put it into a petri dish, and therefore the bacteria can grow. And again I have to put medium out here. So whatever media I'm putting, this media is called as minimal media. Now what is minimal media? Minimal media means the media that contains all the required nutrient for this particular bacteria to survive. Okay, so whatever bacteria survives within the minimal media, this kind of bacteria are called as wild type. So that means they can produce all the things that are required, okay, for nutrient, all the nutrients it can produce, but whatever it cannot produce is present within the minimal media, so therefore they can have a complete growth. Okay, now this wild type is also called as prototrophs. Okay, they are also called as prototrophs. Now there is another type of bacteria. Okay, so suppose I'll just give you two examples. This is one bacteria and this is another bacteria. Now suppose this particular bacteria is a wild type. Okay, so that means it is prototroph. Prototroph means those kind of bacteria that can produce all the required nutrients. Okay, like this is a normal one. Wild type, that means it is found mostly within the environment. This kind of bacteria you'll find. The other type of bacteria that we have is called as the nutritional nutritional mutants. Now nutritional mutants are those kind of bacteria which lack certain kind of nutrient. Okay, these are also called as exotroph. Okay, so what are nutritional mutants? Nutritional mutants are those bacteria which cannot produce a certain kind of nutrient. I'll just give you one example. Okay, suppose this nutrient mutant cannot produce biotin. Just giving an example. Okay, so this cannot produce biotin, so biotin minus, and this one wild type, it can produce everything that is required. Okay, all right. So now if I put both these bacteria within a culture media, okay, and this culture media contains minimal media, that means I'll be giving only those things that these bacteria cannot produce naturally. Okay, so all those food material will be present out here. So what will be the result? When I put this wild type out here, the resultant will be this, that this wild type, they will survive. Okay, so basically if this is my petri dish, okay, not petri dish, okay, suppose this is my culture media, I will see that there is a lot of growth out here, because whatever it cannot make, it is taken up from here, and rest it can make on its own. Okay, now what about this one? This is an autotroph, so it cannot produce biotin, which is they are supposed to make it on their own. Now what happens is, when I grow it, I will see that after some time that the culture media is actually empty. Okay, so media is there, but there is no bacteria, like it's empty, there's nothing out here. Okay, so basically there is no growth. And why this is happening? Because it cannot produce biotin, and without the presence of biotin, these organisms are not going to survive, because it's a important nutrient for them to survive. So this is the difference between prototrophs and autotrophs. Prototrophs, they will take food, whatever they cannot make, they will take it from the minimal media. This auxotrophs will also take, but the problem is that biotin is they are supposed to make it on their own. They don't have this particular gene, so they are called as auxotrophs. So let's look at the definition once. Okay, so nutrient components of the growth media consisting an organic carbon source, that is glucose or lactose, main food source, okay, and various inorganic ions like all this is called minimal media. Now, only prototrophs, that is the wild type bacterium that is able to synthesize all essential organic compound, can survive on minimal media. Nutrition mutants or auxotrophs, bacterium that loses the ability to make one or more essential organic compound due to mutation, cannot survive on minimal media, but grows only on complete media, contains all essential and non-essential compounds. Okay, these are called as auxotrophs. Okay, now let's look at the growth chart. Okay, so this shows the typical growth of bacteria, how it grows. Now, whenever you put bacteria within the culture media, what happens? Initially, they will not grow. Like the number of growth, this is a time, okay, and this is the total number of cells, how it's increasing. So initially, what happens is that there is a lag phase. Like it will take some time to grow. Like this is the time they take to get accustomed to the environment, and there is a lot of stress. Okay, once they have adjusted, they go past the lag phase and they go into the log phase, that is exponential growth phase. Okay, where they will grow in high number, like there will be high number of division, and after a certain stage, when the nutrition like level goes down and all, they'll go to a stationary state where the total number of birth will be equal to the total number of deaths, so that they can maintain their number within the culture media. Okay, so this is how bacteria grows. Okay, now let's talk about a little bit about gene transfer. Okay, so gene transfer, we have vertical gene transfer and we have horizontal gene transfer. Okay, so we have vertical gene transfer and horizontal gene transfer. Vertical gene transfer means transfer of genetic material between members of the same species. Okay, so that means if like for example, if genetic material has been passing on from parents to offspring, that is considered as the vertical gene transfer. Okay, horizontal gene transfer means transfer to genetic material between members of related but distinct bacterial species. Okay, and now if there is distinct bacterial species, then horizontal gene transfer takes place. Now this kind of gene transfer is done by bacterial species through conjugation, transduction and transformation. Okay, so we'll be talking about genetic recombination in bacteria. So basically there are three processes: conjugation, transformation and transduction. So let's talk a little bit about conjugation. Okay, so here I have taken one example. So I have taken E. coli strain K12. This is a strain of E. coli, okay? And these are auxotrophs, that is nutritional mutant grown in complete media. Now complete media means it contains all the nutrients that is required for them to grow. Okay? But they are auxotrophs. Achha, okay, so they are grown. So I have taken two kind of strains. What strain? I have taken strain strain A and strain B. In strain A, this is met minus, that means it cannot produce methionine, it cannot produce biotin, it can produce threonine, it can produce leucine and it can produce threonine. So wherever this plus is there, that means it can produce. Wherever minus is there, it cannot produce. So these are the nutrients that strain A can make and cannot make. Okay, so this is the strain B, the minus one they cannot make and the plus one they can make. So what they are doing, both of them are auxotrophs because they are nutritional mutants, so as they cannot make certain things. Now what I'm doing is, we are taking these two, strain A and strain B, we're mixing it, okay, and putting it into complete media. Complete media means obviously it contains everything, like everything is there, it's complete, it is not minimal. The first one that I was talking about previously, it was minimal media. Only those things that are very very essential for wild type because they cannot produce are only given, like food, okay. And this one is complete media, so it contains everything. So we are putting these two bacteria out here, strain A, strain B, we are mixing it and we are keeping it for overnight. Okay, then what we see, this is strain A and strain B. Okay, I have written the same thing, like the strain A and this is strain B. So what happens overnight, after we incubate it for overnight, the result that we get to see is, now we'll take this and we'll put it into a minimal media. Okay, and again from here also, from strain A also we'll take it and put it into minimal media. From strain B also we'll take it and put it into minimal media. Okay, now when we put into the minimal media, we see that there is no growth. Why there is no growth? Because it is auxotroph and there are certain things that are missing, so it did not grow. Out here also, when we put in minimal media, this is strain B, again there was few things missing, so there is no growth. But out here, what happened? See, I had put A and B together, so some things were present in A, some things were not present in B, some genes were not present. So somehow something happened between strain A and strain B, and they exchanged the genetic material. So whatever what was not present within them, they got it exchanged, and therefore now they have everything. So they could grow in minimal media, but initially the strains separately could not grow. So we got colonies of prototropes. Okay, so that means they have everything what is required. So basically they have everything. Like see out here, everything is plus. So cells grew in frequency of 1 by 10 to the power 7 of total cell. Okay, so what is the inference that we get out of this? The inference that we get out of this is production of prototropes, okay, is a result of genetic recombination between strain A and strain B. Now let's see how does this recombination actually take place. Okay, so how is this recombination taking place? To understand this, a experiment was done where we have taken this kind of tube. It's a glass tube which is called as Davis U tube. Okay, it's a tube, and in center of this tube there's a filter. It's a glass filter with pore size that allow liquid passage but not cell passage. Liquid passage means whatever the media is there, that media can pass through this, but the cells, that means the bacteria, they cannot pass from here to, like, from this through this. Okay, since the pore is very small. So what we did is we put strain A out here and we put strain B out here. Okay, now this strain A contains F plus plasmid. F plus plasmid means, again in the previous video I was talking about this, F plus plasmid means those plasmid that contains the fertility factor, and therefore they can make the sex pilus or the conjugation bridge. And F minus means those bacterial strain that does not have the fertility factor and therefore cannot make the cytoplasmic bridge or the sex pilus. Okay, so what we are doing, we are putting this pressure or suction alternatively is applied so that the liquid can pass through, like it can go from one side to another side, so there is a flow. Okay, so medium is passed back and forth across the filter. Okay, so the medium will go this side and that side depending upon the pressure and the suction that is applied out here. Okay, so here we have put this donor one, and this is the recipient. Because it's minus, obviously it's recipient, it can take up the F plasmid. Okay, so what's happening out here? So when we put strain A and strain B out here, we see that when we plate it after some time in the minimal media, there is no growth. Why there is no growth? The reason why there is no growth is this filter. Now since there is this particular filter through which the cells cannot pass but the media can pass, so therefore what's happening, there is no physical contact between strain A and strain B. And we know that the requirement of the physical contact, that means the cytoplasmic bridge or the sex pilus, is very important. Now since they are not coming in physical touch to each other, so from this experiment we can say, like, whatever was happening out here, we can see like this strain A and strain B, they did not take up things from the liquid, that means the medium, they did not take it. Something happened between them for which physical communication was very much required. Out here, when we are cutting the physical barrier, we are cutting it out, we are not letting the cells meet. In that case, there is no growth. But if we are letting them physically touch each other, then in that case, growth is there. Okay, so the inference that we draw from here is that physical contact between cells of the two strain is essential to genetic recombination for the production of this F pilus, sex pilus, or the fertility pilus. Okay, so F means is fertility factor or F factor. F plus bacteria means they can form a sex pilus and can serve as a donor. Transfers F factor or F minus to the bacteria. And what is HFR? HFR bacteria means high frequency recombination. Again, mutant strain of donor bacteria that undergo deviated frequency of recombination. It is a special case of F plus cells. So this is again two types, F plus and F minus conjugation, and HFR and F minus conjugation. This one I have discussed in detail in the previous video. Okay, so check that as well. And finally, I'll just, okay, last, this one. This is a diagram of conjugation between F plus and F minus. This is also I have discussed in the previous video. Step by step is out there. Okay, so let's recap. So here we have this bacterial chromosome, and this one is F plus, so it contains the F factor. Through this cytoplasmic bridge, what will happen? It can pass. How? So there are two chains in DNA. So obviously one will be cut and it will pass through the sex pilus. It will come to the another cell, and after it comes, replication will start and new synthesis will start. So ultimately this one will also have two strands, and this one will also have two strands, and therefore it's completed. After it's completed, this will be broken, and finally we'll have both F plus X conjugates. Okay, so this was conjugation. If you have any doubt, do let me know.